step, which is you can prime a reaction where if you take two very small pieces of DNA that match perfectly, notice the T matches to the A, I told you earlier that that would happen, and the C matches to the G. I told you earlier that that would happen every time. So I've created these two very small pieces, and in a small way, I've made each of these strands double-stranded just in one area. Double-stranded right there and double-stranded just right there. Okay? DNA has a tendency to want to be double-stranded in the right temperature. And so if you start this reaction, and you start it in the presence of all of these free-floating A, C's, G's, and T's that you see, what happens is this. This. You create two double-stranded pieces of DNA. I just made it an identical copy. And all I did was increase the temperature, add these two little primers, and then lower the temperature. So if I wanted to do the same thing, but I took all these little floating pieces of single nucleotides, single bases, and dyed them, and you can do this, you can dye them, for instance, blue, then the exact same reaction would turn this blue. You'd actually create a blue molecule, halfway blue. And in fact, I went from one copy to two copies. So there it is. Now watch what happens. Right? It's explosive. It's exponential. Every time I run this cycle, I double the amount of molecule that I have. So this is PCR, polymerase chain reaction, because it's like a nuclear reaction. It explodes in volume. I run this 35 times. Each cycle takes 5 minutes. I run this 35 times. It takes an hour and a half, 2 hours, something like that. And I go from one copy to 68 billion copies. And they're all blue. So what does that mean I can do? I guess I'll get to that in a second. First, I want to just maybe look at the economics of this very briefly. When Kary Mullis invented this, he was working for CEDIS. They gave him a $10,000 prize for his creativity, and they built out a patent. And a few years later, CEDIS sold it to Roche for $300 million. And Roche has monetized about $2 billion worth of value from it. And if you ask the average genomicist or molecular biologist, what's the overall industry-wide lifetime value of this thing, they'll say it's more like $200 billion. This one reaction, I can't impress upon you enough, it powers genomics, it powers molecular biology. When you hear about DNA fingerprinting, which I'll talk about in a moment, everything relating to DNA can't be done without this one reaction. And it's happening all over the world right now. We're taking one piece of DNA and we're making 68 billion copies of it. I want to give you one other tool, one other DNA tool, okay, that will complete your tool kit for doing genomic sequencing. And that is scissors. So here is a double-stranded DNA molecule, right? And I'm not showing you all of the letters here or here, but I am showing you this. And if you look carefully at this, this is a palindrome, if any of you know what that means. G-A-A-T-T-C, G-A-A-T-T-C. Now notice, interestingly, I haven't violated my rule. G's are only with C's, and A's are only with T's. So this is a perfectly legal double-stranded DNA. This could happen in nature, okay? Now there's an enzyme. By the way, what's an enzyme? A protein. Everything's a protein, guys. So there's an enzyme called EcoRI, and it finds, its job in life is to find all of the occurrences of this sequence and cut it in this way. Now, you might think, well, how do you make that enzyme? That's what I would think. And it turns out you don't. The enzyme is made by bacteria. And in fact, there's a huge list of these enzymes. You can see here, I'm just looking at the ones that start with A, and they all have different recognition sequences, and they cut in different ways. And the reason why bacteria make this is to fend off viruses. So they're designed, they've evolved to protect themselves from viruses with very specific sequences, to cut viral DNA in the right places so that it can't harm them. It's pretty amazing, right? To me, this is unbelievable. So what we've done is we've decided to co-opt this and use it as a tool. So now, imagine for a second that I've taken some DNA that I want to sequence. And I've cut it, right? And then I've done that PCR reaction, so I've made a whole bunch of copies, and I've made all the copies blue. How do I read the sequence? Molecular biologists, by the way, are very creative. And I'm a computer scientist, and I'm relaying this to you as somebody who's learned this from the outside. And so maybe my amazement is bigger than it should be, but it really blows my mind. Here's what we do. Here's one other thing you should know, which is that the sugar phosphate backbone, the double helix structure of DNA it's negatively charged. You know, it just turns out to have a charge and it's slightly negative. So DNA will tend to walk very quickly to a positive charge, very quickly. So here's what we do. A lot of fancy tricks. We take the DNA, we use PCR to make lots of copies, and then let's say we take some of mine, and we take some of yours, and we take some of yours. And we take each of those samples and we put it right here. That's mine in this little well here, and somebody else's is here, and yours is here, okay? And we just go across all these things. Now, what's going to happen is the DNA is just sitting there in water or some solution. It's just sitting there. But what we do is we create a positive charge down here and a negative charge up here. And so you'd imagine that the DNA would just go whoop, and it would just move really quickly to the positive charge. But what we do instead is we add this very complex gel structure that's really hard for DNA to weave through. It goes very slowly through the gel. It does move, but it moves slowly through the gel. And it moves at a rate which is inversely proportional to its size. The bigger the molecule of DNA, a really big molecule, will move down here through this solution, and ultimately it will find its way, but it will go really slow. And a tiny little piece of DNA will hustle really quickly down here, okay? So with all of these techniques at the hands of molecular biologists, they were able to invent DNA fingerprinting, which is this. Have you seen this kind of a picture before? Some of you probably have. So this is that gel imaged. I'll go back again. Remember, here's each of those lanes, 1, 2, 3, 4, and now if I go forward, here's those lanes again, 1, 2, 3, 4. What I did was I took DNA and I applied one of those scissors, which is site-specific. It's not going to cut once or twice, but probably 5 times or 10 times, because it will find the same site to cut over and over in the genome. So if you close your eyes and think, you'll create small little snippets of DNA, but they'll all be of a certain size. And then you let those walk through an electrical field on a gel, and you get them all pooling. This is a very long piece of DNA, and these are shorter and shorter as you go. Every time I do this with my DNA, using the exact same ingredients in the recipe, I'll have the exact same fingerprint. You'll see 1, 2, 3 things there, 2 there, 1 here, and 1 here. Whereas yours will look different. And this is essentially, it's a fingerprint of the DNA. We don't know what the sequence is, but we know with the same DNA as the starting input, you'll always have this fingerprint as the output. So that was the first idea. Now, watch how you do sequencing. It's the exact same technique, but instead, you take the same DNA. So I just have my DNA now, okay? And I use PCR to make lots of copies, and I dye it. And then I take the same DNA, but what I do is I dye it in 4 different areas. I cut it into 4 pieces, and this piece here, I only dye the A's. So I only am going to make the A's blue. And this piece, I'm only going to make the G's blue. And this one, only the T's blue. And this one, only the C's blue. And when I take that exact same DNA and I run it out, I can tell you that this sequence is G, G, A, T, G, G. You see? So I calibrate this thing so that as it's walking down, you're actually able to measure where the base is left off. And I know that this is a guaranteed 75 base pair, 75 base length for this piece, and this is 72. So there should be 3 bases between, and there are. This is how we do sequencing. Sorry if that was too technical. This is the machine that makes all that happen really easily. It's the ABI 3700, and it is the machine that powered the Human Genome Project. What comes out of that machine is a trace file that looks like this. You look at the gel with the DNA that's moved through. We actually stain the A's red and the C's green, and so each one gets its own color. And then you hire lots of people to stare at this thing and say, All right, there's a blue peak, so we'll call that a C. There's a green peak, we'll call that an A, and you literally go through all 3,000 bases this way. This is the way that we did the Human Genome Project. So we started with this map. We had a rough idea of what the human genome looked like in 1990, and because of a lot of studies that we had done earlier, and we got more and more refined as we went. This is human chromosome 22, and what we would do is simply give pieces of chromosomes to specific labs to work on. And ultimately, we had the moment of success where in 2000, in June or July, these two gentlemen, this is Francis Collins, this is Craig Venter, and you probably know that this is Bill Clinton, came to the White House to say, the human genome has been sequenced. Now in reality, it took them another 3 years for that statement to be true, but it was the way that they could say mission accomplished, if you will. This guy's smiling because he's now the Supreme Director of the National Institutes of Health, and this guy is not because he's not. In fact, the story behind this guy is really interesting. Craig Venter is a genius, and he got so fed up with academia at the height of the Human Genome Project that he created his own company, Celera. And he said, well, we're just going to do it ourselves. And he bought all those machines, and he started sequencing. And for a while, his stock took off because pharma, they wanted access to the data. And they thought, well, hey, I can patent these genes, and I can beat the Human Genome Project to these results. So this guy created this huge enterprise, this enormous thing, until the Human Genome Project was done. At that time, everything that he'd done was worthless because the Human Genome Project is academia, and they just put it out into the public domain. So he created quite a name and a lot of jobs, and then he destroyed that name and all those jobs. And now he's doing something really interesting that I'd love to talk about maybe a bit later, but I don't have the time to get into it now. Briefly, synthetic genomics. What he's doing is he's trying to build life from the simplest amount of genes possible, to create the simplest organisms that he can, and then to be able to add genes to tune them to crack oil or to photosynthesize better or so on. So he's quite a guy. So I said 1988 to 2003, the Human Genome Project, it cost $3.8 billion. How big is the genome? 3 billion, right? Bases. So it cost about $1 per letter. Yeah? And here it is. This is the most important book, perhaps, that we've ever held in our hands. It's the Human Genome. And the only problem is it's written in Chinese, and we can't understand a word of it. It makes no sense to us. It's just a bunch of letters. We don't know the language. We just know the letters. So why is that? The reason why is that we said we sequenced the human genome. Whose genome? Well, it turns out what we did was we averaged about 6 people's genomes. We asked for samples from 30 people, didn't tell them who we were going to choose. We took about 6, and then we went off and we sequenced that. And there's errors in the sequencing, and sometimes my genome will agree with yours, sometimes it will disagree, and we just sort of stuffed it together. And so we don't know anything about the phenotype. So that's a new word that I want to define for you because I'm going to start using it a lot. The phenotype is what it looks like. So my phenotype is I'm short and I'm going bald. My genotype is what my genes are doing, how they are inside of my body, which causes me never to have grown tall and to go bald at such a young age. So the reason why the Human Genome Project produced a book that we can't read is because we only sequenced the average of 6 people. And what we really need to do is sequence enough people to be able to compare phenotype to genotype. So if you put me next to my sister and you ask, well, what's really different about us? Well, she has 2 X chromosomes. I have an X and a Y. But the other thing is she's a little shorter and her hair's a little lighter. And so what you could do, because we're so closely genetically related, you would imagine that our genomes are very similar. And they're only going to be different in a few places. And those few places could describe why we look different, why our phenotype is different. You see? And that's how we can start to understand the genome, is not by doing it once, but by doing it a few thousand times, tens of thousands of times, hundreds of thousands of times. And every time we do it, knowing the phenotype of the organism as well, knowing the phenotype, what they look like, so that you can try to figure out which variations matter. Unfortunately, at $3 billion per genome, we can't do that, right? So, welcome to the Genomic Revolution. And this, by the way, is like last year's news, which is ancient. What you're looking at now is an Illumina HiSeq 2000. It is a new way of sequencing, and I'm going to very quickly share with you how this works. Ready? Okay, here's what you do. You take DNA. You put adapters on either side of the DNA. And then you stick adapters to this big cell here, like this. And then you throw your DNA all over the cell. And then you use this funny thing that I've been telling you about, how single-stranded DNA likes to become double-stranded DNA at the right temperature, and then it opens up again and becomes single-stranded, and you keep doing this over and over, and you grow these clusters. And then you dye each cluster. You throw a wash of these molecules. G's are stained yellow, and T's are green, and C's are blue, and A's are red, and you wash over this whole array just once, and you let it attach one base, one letter to each one of those things. And then you do it again. And you do it again and again and again. And you know, at this particular portion of your array, you've got a sequence which reads G, C, T, G, A. At this one, T, A, G, T, and so on and so on. And you do this maybe a few 100 million times, maybe a billion, something like that. And the reason why you can do it so many times is because this is happening at such a small level, and our ability to manufacture these lanes, see there's 8 lanes here, and in each lane 40, 50, 60 million of these reactions are happening. And so all of a sudden, you've sequenced the entire genome. It takes now about a week. It's going to take about a day by the end of the year. And whereas the Human Genome Project produced 3 gigabases of data, this new way of sequencing produces 600 gigabases. So this Human Genome Project took us 15 years to produce 3 billion bases. Now this machine can run in a week and produce 600 billion bases. You're familiar with Moore's law. That's the idea that the number of transistors on a chip will double every 18 months. And it's another way of saying, roughly, that compute power increases by about 2 every 18 months. And this is maintained for quite some time. So now here's sequencing improvements. For those of you who are mathematicians, Moore's law is an exponent, right? It's a doubling every fixed amount. So this is a far faster exponent, wouldn't you say? The price to sequence the human genome has fallen 100 million times since the Human Genome Project. 100 million times. And it will continue to drop an order of magnitude, at least 2 more orders of magnitude, the way that I see it. Just to give you another sense of it, this is a logarithmic scale. So every time you go up on the y-axis, you're adding not a number, but an exponent. 10 to the 5th, 10 to the 6th, 10 to the 7th, 10 to the 10th. So a line on here is quick growth. It's going up in a regular curve, right? This is world population growth from 1980 to 2010. PC placements in the same period of time. The directory of all scientific literature that's available through the National Institutes of Health in the United States. This is Moore's law. That's the Human Genome Project that you're familiar with. And here's the new stuff. It goes off of the chart straight up, and it's a logarithmic scale. It's unbelievable the amount of data that's being produced. This is a map showing the sequencing machines that have been placed across the planet. You can see there's a lot of density here in the United States. That's where I live. There's plenty happening in Europe. If we zoom into our region here, you can see there's Saudi Arabia. King Faisal Hospital has an interesting hospital. There's some things going on in Qatar. And based on the conversations that I've had with some very interesting people here over the course of the past 2 days, I expect there to be a red dot very soon, right, on Abu Dhabi. China is investing heavily in this technology. The Beijing Genomics Institute has 180 of these machines. This is as of last week. And they, in one lab, have somewhere between 15 to 20% of the planetary sequencing capacity. So if you add all this up, how many genomes can we sequence in a year? Last year it was about 50,000. This year it's about 150,000 genomes, if we turn them all to the problem of human genome sequencing. And that number will go up at a rate which is similar to all of the charts that I've been showing. So our ability to sequence human genomes is exploding. What does it mean for healthcare? So this is a, sorry for the picture. This is a papillary adenocarcinoma. It is a tumor of the glands. And so if you look really carefully, you can actually see it looks a little glandular right there, right? So a 72-year-old male shows up with an itch in his throat. And he says to his doctor, there's something wrong here, I'm not sure what it is. And the doc looks and sees there's something funny, and they do a biopsy. And they find that it's a papillary adenocarcinoma. So what they do is they use a laser to take the tumor out, and they check the lymph nodes. Some of you may know that in cancer, you want to remove the tumor and check the surrounding lymph nodes to make sure that there's no metastasis, no travel of the cancer into those nodes. And they find that 3 of his nodes actually show that there's cancer. So they remove those nodes and they give him adjuvant radiation therapy. So what happens next is he goes away, comes back in about a year, and they see a whole bunch of metastases in his lungs. So this is not normal. This is an adenocarcinoma, and now it's in his lungs. And they do what you do with lung cancer, which is they look to see what kind of a cancer it is. And they notice that there are extra copies of a gene on his genome. And that gene is called EGFR, and it stands, I think, for epidermal growth factor. So I'm not going to get into what that gene does. One of the things it does is helps your skin grow. But if you've got 2 copies of that gene on your genome, the likelihood is you're producing twice as much RNA and you're producing twice as much of the protein. That seems reasonable. Turns out there's a drug called Tarceva, which is designed for cancer patients who have precisely this kind of a cancer, this EGFR positive cancer. So they put this man on Tarceva. And they see him in a couple of months, and everything is growing. It's as if he's had no treatment whatsoever. They don't know what's going on. So at this point, this happens to be a colleague of mine, Stephen Jones, who knows something about genome sequencing, and he says, let's do a genome sequence on him. So they took a sample of his normal genome, and they took a sample of his cancer genome. It turns out that cancer is a disease of the genome. It's a disease where your genome gets massively rearranged, and it allows 1 or 2 cells to proliferate and grow wildly. And so they sequenced both the tumor and the normal cells, the whole genome of each, and said, well, what's really going on here? And what they found was the following. I told you that this patient had EGFR positive cancer. That's true in the DNA. That's true here in the DNA. But when they actually looked at the RNA by doing this whole genome sequencing, there was no difference in the amount of EGFR messages that were getting out. There was no difference. What they did find was this gene right here, which is a tyrosine kinase inhibitor called RET. It was wildly overexpressed. There were way too many copies of this in this guy's cells, in the cancer cells. And lucky for this man, there is a drug which specifically targets this gene. It's called sunitinib, I think. So they start him right here on sunitinib. And after all of this terrible stuff where he shows up with a biopsy, they look at the lymph nodes, they give him the radiation therapy, he has the metastases in his lung, they start the erlotinib, which is Tarceva. He progresses in the setting of that drug, and then they do a needle biopsy, they do the genome analysis here, and then they start on January 1st, 2009, they start this drug. And you know what happens? The tumors shrink. So the use of whole genome sequencing enabled this guy to shrink his tumors. Now, like I told you, cancer is a nasty disease, and it's a disease of the genome. And this drug killed off all of the cells that had this RET disorder, if you will. But cancer adapts. It's like a virus. It changes, and it did so for this patient. And so it stabilized to a certain point. They started another drug called sorafenib, stabilized a little bit, but ultimately this patient ended up passing away due to metastatic cancer. The patient got an extra 3.5 months of life because of a whole genome sequencing. And had that been done at the beginning, who knows what would have happened. Maybe he would never have had the metastasis. This is estrogen receptor positive breast cancer. And this is a breast cancer from a 37-year-old woman. She presents with this, they remove it, they do radiation and chemotherapy, and everything is fine. Except 2 years later, she comes back. She's diagnosed with stage 3C ovarian cancer. They remove the ovaries, they treat with chemotherapy. She goes home. At the age of 42, ovarian cancer returns, even though the ovaries are gone. They treat her with radiation therapy and send her home, chemotherapy and send her home. And then, unfortunately, she comes back about 6 months later with acute myeloid leukemia. And she develops respiratory failure and she dies. That's a lot of different kinds of cancer. To get 3 different kinds of cancer in 3 or 4 years, something is strange. So unfortunately, the patient passed away. My friends at the Genome Institute at Washington University wanted to take a look. And so they sequenced, again, the cancer genome and the normal genome of this patient. And what they found in the